Hi and good morning. We're from Group 6 and our topic for today is control of chromosome duplication. I believe that everyone here knows that chromosome duplication occurs during S phase, which is the phase of DNA synthesis. Chromosome duplication plays a significant role to produce double the amount of DNA. This allows each daughter cell has the correct number of chromosomes after cell division. However, chromosome duplication can only occur once per cell cycle, so there has to be a high-fidelity control system that coordinates chromosome duplication so that it activated only once during each S phase. If not, daughter cells might inherit an excessive amount of any DNA sequence, which could lead to mutation and result in many harmful effects. Now, we will discuss on the control of chromosome duplication. In brief, chromosome duplication takes place in two steps, which are formation of pre-replicative complex pre-RC and DNA replication. Here is the illustration of the chromosome duplication process. Formation of pre-RC occurs during G1 phase. At the start of S phase, activation of SCDK triggers DNA replication to occur. In the following discussion, we will not only further explain on the formation of pre-RC and activation of SCDK under topic cell cycle, but we will also roughly explain on the DNA replication steps under topic DNA replication and repair.
The initiation of DNA replication starts with the formation of the origin recognition complex. The ORC is a heterohexameric complex of 6 protein subunits, denoted as ORC1 to ORC6. Studies in yeast indicates that the ORC binds to a certain region on the DNA strand called the origin of replication, in which specifically contains high concentrations of adenine and thymine nucleotides. ORC1 to ORC6 have conserved AAA plus folds and characteristics of ATP-dependent clamp-loading proteins, which allow ring-shaped protein complexes to encircle duplex DNA. ORC1 to ORC5 are required for origin recognition and binding, while ORC6 is essential for ORC DNA binding.
Next, I will explain to you about the formation of pre-replicative complex, pre-RC. Firstly, the origin recognition complex, ORC, binds to the origin DNA. ATP binding is required for assembly of a stable ORC and can stimulate DNA binding. Then, cell division cycle 6 or CDC6 is recruited to form an ORC-CDC6 complex on origin DNA. The minichrome maintenance 27, acronymically MCM27, which has a function as a DNA helicase, will form a complex with the chromatin licensing and DNA replication factor 1, acronymically CDT1 protein. This MCM-CDT1 complex is recruited to ORC-CDC6 complex, thus forming pre-replicative complex, pre-RC. ATP hydrolysis is required for MCM loading and the release of CDT1 protein. Then, CDC6 will be dissociated from the chromatin to trigger the replication. So, once MCM27 is loaded, it no longer requires CDC6 or CDT1 to maintain its origin association.
Once pre-replicative complex pre-RC is formed, cell cycle control system continues with the activation of cyclin-dependent protein kinases, CDKs. At the start of S phase, SCDK is assembled around the DNA strand in inactive form. Then, SCDK activated by its regulator, which is S-cyclin, to form S-cyclin-CDK complex in partly active form. Full activation of the cyclin-CDK complex occurs when CDK activating kinase CAK phosphorylates an amino acid near the entrance of the CDK active site, causes a small conformational change. This increases the activity of the CDK. S-cyclin CDK complex is in fully active form now. In doing so, S-cyclin CDK complexes essentially stimulate and initiate DNA replication in S phase.
During S phase, SCDK activates the DNA helicases in the pre-replicative complex, pre-RC, and promotes the assembly of the rest of the proteins, such as MCM2 to 7 helicases, to promote formation of replication fork. Initiation step will be the first step in DNA replication. During initiation, binding of the DNA helicase to the origin of replication will be formed. DNA helicase will disrupt the hydrogen bonding between the base pairs of the two DNA strands. It is important to separate the DNA strands into a Y shape, which is known as replication fork. This area will then be the template for replication to begin.
The leading strand is the simplest to replicate. In the initiation process, the DNA polymerase can start making a DNA chain from scratch. It requires a pre-existing chain or short stretch of nucleotides called a primer. Once the DNA strands have been separated, primase bind to the single-stranded DNA to generate RNA primer. It provides a 3' end for DNA polymerase to work on. A typical primer is about 5 to 10 nucleotides long. The primer always binds as the starting point for replication.
The third step of the DNA replication is the elongation. Enzymes known as DNA polymerases are responsible for creating the new strand by a process called elongation. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria such as E. coli, polymerase III is the main replication enzyme, while polymerase I, II, IV, and V are responsible for error checking and repair. DNA polymerase III binds to the strand at the site of the primer at the beginning. DNA polymerase III moves from 5' to 3' direction and adds new base pairs complementary to the strand during replication. In eukaryotic cells, polymerases alpha, delta, and epsilon are the primary polymerases involved in the DNA replication. Because replication proceeds in the 5' to 3' direction on the leading strand, the newly formed strand is continuous. At the same time, multiple primases bind to the lagging strand to form RNA primers. After these primers are formed, the primases detach from the lagging strand. The lagging strand begins replication by binding of DNA polymerase III to the RNA primer. Each primer is only several bases apart. DNA polymerase then adds pieces of DNA called Okazaki fragments to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjointed.
So the final step is termination. Once all of the bases adenine are matched up with thymine and cytosine are matched up with guanine, new DNA are formed. Next, an enzyme called exonuclease will bind to the RNA primers at the start of each leading strand and Okazaki fragments. This will strip away primers and also leave gaps. After that, DNA polymerase I will bind to the strands to replace the gaps that were initially filled with RNA primers with DNA nucleotides. At this step, the new strand is already proofread by DNA polymerase I to make sure that there are no mistakes in the template strand. If errors are found, this will lead to incorrectly coded gene. This can likely to be destructive for an organism. Luckily, this error only happens about once in every 10,000 base pairs. Finally, enzyme called DNA ligase bind to each strands to seal up the fragments of DNA, forming a new strand that is one long continuous DNA strand. DNA replication is described as semi-conservative as 2 copies from each new DNA molecules is made of one strand from the original DNA and one from the new one. Following replication, the new DNA automatically winds up into a double helix. That's all from us regarding our topic, control of chromosome duplication. Thank you.